thank you all for coming, everyone. So you know that this is the second international speaker for our series, and I'll introduce him shortly. Obviously, the aim of this seminar series is to stimulate collaboration and communication between people within the RadNet City of London Centre, people within RadNet centres across the country, and people obviously interested in radiation oncology and radiation biology generally across the globe. So these talks are open to people from all institutes. So please do spread the news about them to your colleagues. You know, we want to have as a wider audience as possible. We are 2 minutes past, and we still keep getting beeps of people coming in. But I will start introducing the speakers. So obviously, my name is Crispin Hiley. I'm a clinical academic at UCL. It's a great pleasure for me to introduce Mohamed Abazeed. He will be the second international speaker for our seminar series this year. He's a really excellent radiation oncologist and physician scientist. He uses transomic approaches in order to improve the personalization of cancer treatments. And as some of you who attended the talk I gave yesterday will know, he's obviously doing a lot of work to try and see if we can get past a one-size-fits-all for radiation dose for patients. He's made some really great contributions looking at how tumor mutations predict therapy resistance, and in particularly radiation. And he uses both bioinformatic as well as genetics and functional genomics approaches to look at this. He did his MD PhD at the University of Michigan with research into genetics. And he did his residency training at Harvard Radiation Oncology Program, where he was the Leonard Holman Research Fellow and conducted his research at the Broad Institute, which many of you will know as a global leader in cancer genomics. And he's currently now Associate Professor at Northwestern University's Feinberg School of Medicine. He's the Scientific Director of their Lung Cancer Program and the Inaugural Director of their Center for Precision Radiotherapy. So thanks very much, Mohamed. We're really looking forward to hearing your talk.
Thanks, Crispin, and great to see you and my European colleagues. It's been quite a while since we were in the French Alps together, and I remember those times fondly. It seems so distant. Okay, so today I have a wonderful audience. I finally get to talk to a bunch of radiation oncologists and radiation biologists, and I don't have to be ashamed of that. We're going to talk about the lab's efforts, some interrupted, but now kicking into high gear, hopefully, about answering some of the questions of the genetic determinants of response to radiation. So Crispin, if you don't mind, so I can share my screen. Excellent. Okay, so the lab that I started in 2013 has been heavily focused on incorporating multi-omic approaches to predicting response to radiation. We're going to talk about the computational, the experimental, as well as the potential clinical trial difficulties of this line of work and how we're trying to advance that using advanced translational models in the laboratory. So the focus of this presentation is really going to be on gene mutations with some reflections and some thoughts about future directions using PDX program, as well as clinical incorporation of data. Okay, here are my requisite disclosures. So how do we think about personalized medicine from the perspective of the radiation oncologist? In many ways, our medical oncology colleagues have it much easier, right? There is a single gene, single drug addiction model to cancer that has made biomarker identification in solid tumors substantially less computationally rigorous and challenging. The idea of stratifying patients on the basis of these significant driver mutations that are mostly clonal have really changed the way medical oncologists have approached their patient populations. Radiation oncologists have yet to adapt to this, and there are multiple limitations to this. The first is the translational research infrastructure isn't well established in most radiation oncology academic centers. But beyond that, it is not a single therapeutic single gene phenomenon. It's in fact multigenic. So the response prediction difficulties are substantially more complex than they are for single gene, single phenotype. So, but even in targeted therapies, even when using TKIs, for example, in EGFR mutated lung cancers, there is significant complexity of the tumor, and the tumor has adaptive capabilities. And so we also have to be cognizant of that. So in tumor systems, I tend to think a lot about this from a complex system biology perspective. Tumors are really adept at maintaining their generative capacity. And so when stressed from any perspective, the network can respond, and it typically responds by either changing the ability of the drug to penetrate the cell by multi-drug resistance, by inducing a mutation in that gene and blocking the entrance of the drug to the inhibition pocket, or alternatively by activating a pathway that can circumvent resistance. And we see this in EGFR inhibition by MET amplification, T790M mutations. The same is likely to be true for radiation, and we'll talk about intratumoral heterogeneity and how resistance can evolve over time. So we think of the cells as greater than 27,000 transcripts with gene networks that are disrupted by gene mutations. So there's incredible complexity and redundancy in the network. In the case of radiation, we're not really talking about tumor response. Most tumors respond, but for radiation, really the benchmark of success is persistent and longitudinal local control. You know, how do you define local control in cases in breast cancer, for example, in which you can have delayed intratumoral breast recurrences 8 to 10 years after? And so durable local control is really the clinical benchmark and also the translational benchmark that we have to assess. If a few clones survive, that leads to tumor repopulation. And so it's much more challenging for radiation oncologists to identify biomarkers of local control than it is to simply identify biomarkers of tumor regression or response. The systems are substantially more complex as we're beginning to learn. There are tumor-associated fibroblasts, there are immune cells. This is an open system with tumor-infiltrating lymphocytes potentially circulating. And so how do you factor all of these different components into your prediction model? And then one of the last features, which I think is really critical, both on the genomic and the epigenomic scale, is the dynamic nature of tumor response. So tumors have substantial intratumoral heterogeneity. This is a subclonal reconstruction of a head and neck tumor in which you have two distinct subclones. After irradiation, this tumor recurs, and really the main determinant of this tumor was this subclone. This isn't representative of all head and neck cancers, and in many other cases, new subclones that have a distinct identity from pre-therapy subclones emerge. So really understanding longitudinal resistance is key. But we have to start somewhere. And so we have to build scaffolds that allow us to be able to study these phenomenon, not just using snapshots in time at tumor diagnosis, but potentially longitudinal translational models that allow us to understand the dynamics of resistance. So our lab has expended a substantial effort previously in Boston, then in Cleveland, and now here with our program getting set up. Which is really critical for us is the use of patient information and patient data to really drive a significant number of our questions. And so this requires a tissue and blood bank resource. It requires extensive analysis of clinical data, EHR, with a significant program here in natural language processing and extracting in an automated fashion how well patients are doing or not doing using automated scanning of pathology reports and clinical notes. I won't talk about our imaging work, but that has been a robust part of our program in the integration of imaging with clinical and histopathological information. I'll be mostly focused on the translational component here, which is the use of cell line derivatives as well as PDX models in order to formulate models that can predict. And what are we trying to predict? Well, we're trying to predict local control as a function of time, and that is it's not a binary. It's not useful to predict whether a patient is going to die or not. It's quite useful to predict when a patient is going to die. And so the same goes for local failure. We'd like to know the time to local failure, so incorporating time to event predictive models is really key. So I'll talk briefly about our previous effort that used the cancer cell line encyclopedia, 533 cell lines. This was the largest profiling effort for radiation survival to date. And some of the genetic associations that were made, we think this was a nice contribution, but we needed to improve upon this contribution because it largely focused on associations rather than causative, rather than determining whether the mutations that were associated with resistance and sensitivity were actually causing resistance and sensitivity. And so we initiated Radiogenomic Atlas, which has now tested more than 600 variants in over 1,200 replicates using a arrayed platform, systematic unary testing, so single variant, single cell. And then our restarting of our PDX-ray effort, we've generated close to 361 PDXs, and this has now been relaunched in the summer of 2020. And we'll have more data for you there. We've now profiled close to 25 PDXs, and we've learned a lot about how to do this in high content fashion. So the Cancer Cell Line Encyclopedia is a wonderful resource, and it now includes more than actually 1,200 cell lines with a significant number in lung, incidentally, and lots of solid tumors represented, so over 34 cancer subtypes. And this is a great resource for the literature, has been the source of multiple multi-omic publications and increasing omic inputs, including now DNA methylation as well as ATAC-seq data. So this was a wonderful resource. In 2013, we initiated the XTDT squared project, which profiled the 500 or so cell lines. We used a high content platform that closely mimics the colony formation assay. And what we showed was substantial correlation with the CFA and variation between within and across cancer subtypes, with mostly Gaussian distributed radiation survival as measured by area under the curve. So this suggested what most of us clinicians long suspected, which is that there's significant variation both within and across tumor types as it relates to radiation sensitivity. However, one of the limitations of the original publication was that we were able to identify genes that were highly associated with resistance, and in this case, I'm showing sensitivity. We were able to draw some general remarks about the potential role of these genes in DNA repair, for example. However, what we really couldn't identify, which is critical for clinical guidance, was whether individual mutations actually conferred the phenotype, or rather than simply being accidentally associated with that phenotype. There was also the limitation of identifying genes as a function of whether a mutation occurred in the gene categorized in the binary of 1 or no mutation in that gene of 0. Genes are large and have structured domains and unstructured aspects that link those domains. And so looking at individual domains was not something that we did in the original publication. So we really spent a lot of time computationally thinking about how to enrich for selection of variants that may be functional using UniProt in PDB domain guidance. And I'll show you examples of that shortly. So how do you take all of this information? You have the original phenotype genotype data from XTD squared, otherwise known as our cell line profiling effort. And you also, in the meantime, have substantial amount of computational development, including things like mutation assessor, which can measure the saliency or the likely functional outcome of a specific mutation in a gene in or outside of a particular domain. So we wanted to integrate all of this to generate a candidate nomination strategy for the identification of mutations that, when profiled, could potentially alter our phenotype. And so we approached this in two ways. We enriched on the basis of whether genes that were mutated were associated with resistance or sensitivity using the original cell line profiling effort. And then we conducted the analysis incorporating UniProt domain, whether a mutation occurred in the UniProt domain or not. And using this binary computational output, we then looked at associations using our information similarity metric, and we were able to make some computational predictions whether individual mutations in individual domains were predictive of resistance or sensitivity. And then that is what gave us our list of 600 genes. This was complemented by or supplemented with commonly mutated genes. So if genes like KRAS, for example, or P53 were not nominated by the strategy, we still wanted to assess it. So that's the rationale for augmenting with commonly mutated genes. The experimental strategy is as follows. Again, there are multiple computational resources that can predict whether a variant is likely to be gain of function, change of function, or loss of function. So on this basis, for genes that were predicted to be gain of function, dominant, negative, or change of function, we were simply able to integrate that using a lentiviral vector. The cell line of initial use is BS2B, which is an upper airway cell line that is immortalized with an SV40 virus. The SV40 leads to a mild diminution of P53, but in addition also decreases RB1 function. So by integrating this gain of function, it would express its phenotype, hopefully, and then we can assess its effects on radiation survival. In cases in which we thought that the genes, or we predicted that the genes were loss of function, we clearly needed to devise a distinct strategy. We needed to knock out the loci in our immortalized and potentially cancer cells in downstream testing. And once you integrate this, if this is an integrant that is functionally silent, it will complement the phenotype. If it is loss of function, then you will be able to assess that and measure radiation survival in that context. The approach here to be able to do this in high content fashion and only knock out the gene a single time and allow us to use our really vast library of lentiviral variants is to use this junction CRISPR approach, which targets the exon-intron junction of the gene of interest. Because the integrated ORF lacks introns, obviously, the CRISPR, although expressed, will not target the ORF, and this will allow you to express your protein of interest while knocking out the endogenous copies of the gene. So how do we interpret our profiling results in cases in which no knockout was conducted and you were to express a gain of function allele that conferred resistance or a dominant negative allele that conferred resistance? You would see enhancement of the phenotype. If these alleles did not cause resistance, then it would simply fall near the control curve. In cases in which there were cases in which we knocked out a gene, so this is the baseline. You knock out a gene and it caused radiation sensitivity. So think of your DNA repair genes, for example, P3BP1, which I'll show you later. If you were to put back a gene that was functionally silent, you would simply restore the function of let's say, P3BP1. If you were to integrate a loss of function allele, it would have sensitivity profile very similar to the knockout. So this is a way of distinguishing functionally silent from loss of function genes. And then lastly, the other scenario is if you knock out a gene and it causes radiation resistance. KEAP1 is a great example of this, which activates the NRF2 pathway. If you were to put back a KEAP1 variant that is functionally silent, it would restore the function just like a wild type. However, it was loss of function, it would not be able to restore sensitivity. So I know there's been a lot of confusion about our interpretation, so hopefully that clarifies it, and I'm happy to answer any question in the question and answer session. So this is the latest update of our profiling effort. We used NRF2E79K. This was identified as one of our strongest predictor of radiation resistance from multiple experiments in our laboratory that span back to when I was a postdoc. And so this was our benchmark for resistance. We also knocked out P3BP1, and this was our benchmark for sensitivity. So that gives you sort of the dynamic range of radiation survival. There are the initial part of this effort. You can imagine knocking out genes that are involved in DNA repair can be quite challenging, and complementing them back. These are usually 5 to 6 kb genes, and so that part of the process is still ongoing. However, so that's why you see most of the initial alleles is focused mostly on the resistance side. However, there are some interesting observations. So first of all, other NRF2 alleles scored quite highly, which is a nice sanity check. KEAP1 alleles, these are now dominant negative KEAP1 alleles, not in a knockout setting. And so we confirmed some KEAP1 dominant negatives that scored very nicely. We have published some work on BRAF that appears to score. There are other genes that are less known. For example, DRD2 and CYP5R4. The knockout of CYP5R4 induces radiation sensitivity in work recently published in Cell. So the overexpression of this gene appears to confer resistance. And additional genes that I'll talk about in greater detail. I want to draw your attention to p53, VHL, interestingly, NRAS and KRAS, which did not score substantially, at least when looking at the full extent of the dose response curve. But we'll come back to that. So how did the previous effort help us, and how did our enrichment effort using mutation assessor and other PDB enrichment, the protein domain enrichment computational approaches facilitate the identification of variant alleles? So if you were to take all of the alleles and split them by incidence by quantile and TCGA, you will see that if the incidence is higher, meaning that there's strong selection for that allele in human tumors, you see a substantial induction of resistance. If you were to compare our XTD squared effort with our Atlas effort, so cell line profiling, you see a general trend. So if they scored on the low end in XTD squared, or they were associated with sensitivity in XTD squared, they tended to score lower by Atlas. Similarly, or the converse of that is also true in terms of the highest quantile in XTD squared. Those really were the alleles that conferred resistance in our cell line profiling. The in silico hit rate was substantially above commonly mutated genes and genes that were selected in which we saw very little association with radiation resistance or sensitivity. And we defined a hit as being within a standard deviation of NRF2 E79K and P3BP1 knockout. There were several questions about doing this and the importance of cellular context. Why pick BS2B? Why pick an immortalized cell line? Why select this particular type of immortalization? The answer to that is obviously that's an exigency. It's a practical matter. To do this in multiple cell lines would have been very challenging at this scale. However, one can now take alleles that span the survival spectrum in BS2B and test them in distinct cellular context. So NCI H520 is a lung cancer cell line, a lung squamous line. And so what you're seeing here is a scatter plot of survival of multiple variants, close to 32 in these two distinct cellular contexts. So similar lineage, both lung, one is a cancer, one is an immortalized cell line. And what you see here is essentially imagine throwing darts at a dartboard. If they were resistant to BS2B, they were more likely to be resistant to H520. And so the enrichment above the expected 25% ratio is shown in quadrant 1. This appears to hold for a immortalized bladder line. The immortalization process here is using hTERT, which affects telomere length. However, there was less of a consistent pattern in the bladder cell line. There were some interesting outliers in terms of sensitivity, and we're certainly interested in that. So cellular context appears to be important in some settings, but for the most part, cell variants that conferred resistance in one immortalized cell line or cancer cell line appeared to confer resistance in other cellular contexts, with some interesting outliers. We were interested to see how much can this inform chemotherapeutic response. We had shown in our previous effort that response to radiation mimicked in terms of the sensitivity profile responses to chemotherapeutics, with an emphasis on therapeutics that affected DNA double-strand breaks. And so we were pleased to see, for example, VX680, which is a mitotic spindle poison, affects aurora kinase and causes G2M mitotic catastrophe, similar to radiation, was highly correlated with radiation response. That is, variants that conferred resistance to X-rays also conferred resistance to VX680. And that seemed to be the pattern for most chemotherapeutics, with some exceptions, which I'll show you shortly. This is a principal component analysis of drugs and radiation, with radiation being at the center. And so what you'll see here is drugs that are often used with radiotherapy: carboplatin, etoposide, docetaxel, that were very close on this two-dimensional principal component analysis, suggesting that they had similar genetic dependencies. There were other drugs that fell far away from that, including bleomycin and methotrexate, which we'll come back to shortly. Some alleles conferred pan-resistance to both radiation and chemotherapeutics. These are the alleles you see here. Others had a mixed effect on radiation and chemotherapeutics, and others appear to be selective for radiation, suggesting that this isn't a panacea, that there are some alleles that are going to be selectively resistant to radiation, and some that will confer resistance to both traditional chemotherapeutics as well as radiation. Bleomycin and methotrexate had an interesting pattern. They actually, in contrast to other chemotherapeutics, they actually induced sensitivity in variants that cause resistance to radiation. Specifically, I'll focus here on bleomycin. We think we understand the mechanism of how this is occurring. So in bleomycin's case, so this EGFR caused resistance in our platform, with high expression of EGFR with L858R, as well as the T790M mutation. Both of these alleles were quite sensitive to bleomycin, and the mechanism has been previously worked out by other groups. But essentially, the higher the glutathione levels, the more rapid you can reactivate bleomycin and cause more DNA double-strand breaks. So we think we've identified a dependency. So in tumors that potentially could be resistant to radiation, the use of bleomycin in the adjuvant setting, although there is a risk of radiation pneumonitis in the lung, can certainly be considered. We have an interesting story developing with methotrexate and its effect on methionine levels and methionine funneling to glutathione using a process called transsulfuration that we're not ready to talk about yet, simply because we just need a few more experiments to validate before I feel comfortable talking about it. There were some interesting patterns regarding the dose per fraction. And so when our platform measured doses at 2, 4, and 6 gray. When you compare the distinct doses, they're mostly highly correlated. So whether they were sensitive at 2 gray, 4 gray, 6 gray, or 4 gray, really strong correlation coefficients above 0.9. However, when you compare 6 gray to 2 gray, you begin to see some outliers emerge. And when you filter out the NERV2 and KEAP1 alleles, what emerges really are KRAS variants. Now, interestingly, if you remember from the original profiling figure that summarized all of our variants, KRAS did not score substantially. And however, when breaking it down by dose per fraction, you see a significant enrichment for KRAS. So we wanted to confirm this, so we actually took multiple KRAS alleles and reinfected them to validate that this in fact was taking place. What you see here are G12, G13, and Q61, A59, and E62, which are associated with the P loop, the switch. So the color pattern here is here to help you in the switch 2 domain. And what this data suggests is that there's really no difference at 2 gray per fraction compared to expression of KRAS wild type. However, at 6 gray per fraction, you begin to see an induction of log-fold resistance, with perhaps an increased level of resistance for these less common Q61 alleles, which occur at less than 1% frequency in non-small cell lung cancer. So, as we were collecting all of the cell line data, we obviously wanted to correlate it back with clinical outcomes. Our former institution, unfortunately, did not have a robust genomics pipeline, and so most of the patients were not subject to panel sequencing. We're very fortunate here to have a pretty robust genomic pipeline that dates back to 2014, with initially KRAS and EGFR sequenced in almost every patient. In 2016, 22 gene panel sequencing that includes some of the most commonly mutated genes in lung cancer. And in 2018, that's now been expanded to 202 genes. So there's actually a very nice clinical correlate data set that has emerged that may allow us to investigate some of the associations with our cell line data. So the confounder, of course, with radiation is frequently used with chemotherapy, and whether concurrently or in the adjuvant setting. So focusing on stereotactic body radiation, which is a focal form of radiation that is typically used alone, and if chemotherapy is given, it's usually given in the adjuvant setting. We filtered out any patient that received systemic therapy after radiation. So this is a really clean data set of 197 patients, with more patients that we're annotating. We think we can get up to 500 patients that have received radiation alone. So we think it's a pretty unique resource. So because KRAS is a commonly mutated gene, and because we've been sequencing KRAS since 2014, we actually have now annotated 95 cases in which KRAS was sequenced, and we've given radiation alone to patients. So we can actually test whether this in fact might be true. So for those of you not familiar with stereotactic body radiation, it's typically used for early stage lung cancer, as well as oligometastatic disease. These are patients that have received thoracic radiation exclusively. And so this is an example of a KRAS mutated early stage lung adenocarcinoma that was treated with, and this is its FDG avidity on PET scan. Prior to therapy, it was treated with SBRT, 50 gray in 5 fractions. There was an initial response, but then there was regrowth. And this regrowth was confirmed by an increase in PET. This regrowth occurred at 14 months. And so we define local failure here, which is another really important element of clinical integration, as persistent growth. And so the confounding element post-radiation in the lung is that it can cause a pneumonitis and fibrosis, which can confound the interpretation. And this fibrosis can evolve over time. That can confound our interpretation of whether a tumor truly failed. And so in 50% of the cases, we actually have biopsy confirmation of failure. In others, we've had to rely on persistent growth in addition to an SUV max that supersedes that of the initial scan at diagnosis. So using this data, we can hopefully clearly see that in patients that are KRAS mutated, this includes early stage lung cancer patients exclusively. KRAS mutated patients have a substantially higher risk of local failure, with a hazard ratio of 2.23 and a p-value of 0.02. The dose per fraction here, the median was 100 gray per fraction. Historically at this institution, the incorporation of higher doses, for example, 54 to 60 gray in 3 fractions, hasn't been that common. That is changing now. And so, please take this interpretation with the view that the vast majority of patients received the lower end of the biologically equivalent dose, which is 100 gray. There were some patients that received SBRT doses for palliation purposes and survived long enough for us to track them, but the vast majority received about 100 gray per fraction by BED. The alleles in KRAS, we weren't powered to detect any specific allelic differences. The vast majority of the alleles are in G12, with secondarily in G13, and again, very rare that you would find a Q61. We did find one case that was Q61, which incidentally recurred, but that's simply anecdotal. This is an interesting space for multiple reasons. But actually, before I get to that, so Max Dean has recently published on NRF2. By the way, we do see a NRF2 association. I'm just not showing it here. Max beat us to it. Max Dean's work showed that the correlation with the genome was really negated by tumor volume. We are not seeing that for KRAS mutated tumors. CT on axial diameter is actually a little bit less. This is not significant in KRAS mutated tumors than wild type. So Max's interpretation was that the genome wasn't driving resistance, so in fact, tumor volume, which we know in SBRT is a driver of resistance. But this is clearly a genetic determinant of resistance. These tumors are smaller and still failing at a higher rate. There is a lot of interest in KRAS, as most of you probably know. In ISLAC a couple of weeks ago, there was a report from the Amgen compound that targets G12C. There are other drugs in development by Mirati and others that are targeting G12C, but in addition, G12F and G12A. So we really think that this could impact clinical management clearly either in the neoadjuvant or the adjuvant setting. The use of KRAS inhibition can potentially mitigate these local failures if dose is limiting sort of for non-peripheral lung lesions. We also think that these local failures, although we're not powered to detect it, we're seeing local failures are predictive of nodal and distant failures. So hopefully, as our clinical data set increases in size, we'll be able to show that local failures really do matter in early stage lung cancer. So let's talk about categories of mutations within genes. Some of this work has been nicely laid out by Neil Hayes at UNC, by Matt Meyerson and Peter Hammerman in 2013 with the lung TCGA squamous cancer publication, and more recently by Max Dean's group that looked at 57 patients who received stereotactic body radiation and showed an association of NRF2 and KEAP1 with resistance. I think one of the critical points for alleles that are loss of function is that they really can't be categorized into discrete bins. So what we did here is we used our junctional CRISPR approach to knock out KEAP1. This is baseline KEAP1, knocking out KEAP1, KEAP1 plus vector control, and KEAP1 plus wild type. So you can see the system actually works quite nicely using the junctional CRISPR. It doesn't recognize this ORF, and you're able to get stable expression of your protein of interest. And so phenotypically, what you see is here's non-targeting control. The knockout of KEAP1 induces resistance to radiation. You add back a vector, nothing happens. You add back wild type, and you restore sensitivity to baseline levels. So this is a nice complementation assay to look at loss of function alleles in KEAP1. And so that's what Dr. Gopal did here, is she took variants of KEAP1 and expressed it in a KEAP1 knockout background. Here's what two knockouts of KEAP1 standard deviation looks like. And here are multiple replicates of KEAP1 wild type. So if a gene that carries a variant is expressed and restores the sensitivity to the resistance induced by the knockout, a lot of double negatives here, then that allele is essentially mimics wild type. So it is functionally silent. It is not loss of function. It's not gain of function. If the allele is put back and doesn't completely restore sensitivity in a KEAP1 knockout setting, then by definition, it has some loss of function, assuming that it's equally expressed, of course, which, of course, we're confirming. If an allele that is put back into a KEAP1 knockout setting causes more sensitivity, then it is hypermorphic. And you might say, well, how on earth does that happen? This actually has been described very nicely by Neil Hayes' group at UNC. These are KEAP1 superbinders. So this is a confirmed superbinder of NRF2. KEAP1 is a scaffold protein that binds to NRF2 and degrades it. NRF2 is an important transcription factor that is involved in oxidative stress response. So its activation leads to therapeutic resistance. By these alleles binding tightly to NRF2 and degrading them, they can actually induce a phenotype above and beyond the knockout of KEAP1. And so using this complementation system, you can nicely stratify alleles into at least some murky categories of a function, hypomorphic, wild type, and hypermorphic. Now, this is a continuum, and I think that's one of the points here is, our medical oncology colleagues really have to rely on mostly biomarkers that are binary. You either have an ALK translocation or you don't. You either have a MET exon 2 skipping mutation or you don't. And that's how they select their therapy. And their therapies are essentially single dose. So you either have high dose chemotherapy, low dose chemotherapy, or drug or no drug. So it's a binary selection. However, we're very fortunate in that we can calibrate our dose at really to decimal levels of precision. And so really understanding how these distinct alleles affect radiation survival can inform our prediction models so that we can tune radiation appropriately. The next important step for us is obviously clinical tumors are much more complex. They're typically driven by one or two drivers. They typically have a p53 mutation or some other tumor suppressor mutation. So it's not as if the combinations are infinite. Of course, they're finite. However, there are interactions that we are missing using this unary testing approach, which is, again, single variant, single cell line, and measuring radiation survival. So we've moved into the next phase of this project, which is to really begin to assemble and understand the interactions between different genes. And I have one vignette here that will hopefully illustrate why we think that this is going to be an important part of the next phase. And so this is lung adenocarcinoma by TCGA. And these are the most commonly mutated genes in lung adenocarcinoma. BS2Bs are mostly an adenomatous cell type, which is why we selected lung adenocarcinoma as our initial analysis pipeline. So some of the most commonly stratified by Q value, some of the most commonly mutated genes in lung adenocarcinoma include STK11, p53, KRAS, and KEAP1. We're really interested in potential interactions across these most commonly mutated genes. And so we did a mutual exclusivity analysis, and we found that among the most commonly mutated genes, STK11 mutations and KEAP1 mutations are most likely to co-occur. So the cancer prefers at an odds ratio of 1.7 to have both of these mutations mutated. That may suggest that they are activating complementary pathways to drive oncogenesis. And the question that we queried was, is this combination also promoting resistance to radiation? So we have two different guide RNAs to STK11, and we used our KEAP1 allele from the experiment that I just showed you. So you knock out STK11, nothing really happens as it relates to radiation resistance. This guide RNA had a substantially better knockout than this. So there's some residual STK11. I'm sorry, I don't have the Western blot to show you here. When you knock out KEAP1, you induce resistance as you would expect. Then the combination, you see an enhancement of resistance, and here substantially more impressive. You see almost a doubling of resistance in the context of knocking out STK11 and KEAP1 compared to KEAP1 alone. So this suggests that this propensity for co-occurrence in cancer also leads to a more enhanced radiation resistance. So we think using this information capability, we think this can guide our clinical association studies and certainly can improve our pretest probability of identifying associations that are more likely to be biologically plausible, but also more accurately predictive of response to radiation clinically. So with that, I'd like to conclude and thank the many members of the team that contributed to this that spanned multiple institutions. I'd like to thank Brian Yard, who initiated this effort. Brian is now at Viocera, which is a small startup company in St. Louis. Brian and Aaron really did a lot of the heavy lifting in terms of the creation of the libraries and the initial testing pipeline. Jess Castrolon, who was a graduate student at Case Western Reserve University and really contributed to the drug analysis and interpretation of the drug analysis data. Titus and Will Yang. Will was a rotating graduate student in our laboratory. Titus Barrow is a technician who's now in medical school, but did a fantastic job over the summer to helping us get to where we are today. And lastly, I'd like to thank Dr. Gopal, who's taking the lead on this project, and a lot of the validation studies you saw today come from Dr. Gopal's hard work on this project. I'd like to thank our funding sources, the American Lung Association and the NIH, as well as ASCO, and our collaborators who we've been able to maintain across multiple cities now. And with that, thank you so much for your attention. I'd be happy to answer any questions that you might have. And it's been a pleasure chatting with you guys.
Thanks very much, Mohamed. That was a great talk. As ever, you always manage to make what is an incredibly complicated topic seem accessible. So please, if anyone's got any questions, please do put them in the chat. If you're too nervous, or raise your hand and you can unmute and ask. I mean, I'll ask the first question here from the chat, which is, in your cell line screen, I think they were asking about where the cell lines came from. Obviously, you're using Bs2Bs, but I guess they're sort of thinking about cell lines that might come from other sources, sort of from patients, or primary versus versus metastatic, and previously treated versus untreated patients. I mean, I guess if you, I guess that kind of leads to the genomic context of where these variants are, which is what you're talking about. Have you got any thoughts about that as a comment/question?
Yeah, so obviously the cell line derivatives that were created historically were mostly from metastatic patients. In the cases of lung, these are largely pleural effusions. So if you go back and look at some of these derivatives, most of them developed at the University of Texas Southwestern, specifically for lung squamous cell lines, mostly in the metastatic. So these are derivatives; they're representatives of the original primary tumor. There's, the original CCLE publication showed that these cell line derivatives do maintain some genomic fidelity to their primary tumor analogs. However, you do get a lot of genetic drift. So think of these as representatives of tumors, but certainly do not represent the full transcriptional and genetic intertumoral heterogeneity that exists in primary tumors.
Okay, that's great. I've got a couple of questions while hopefully some others are thinking of some. So I guess, slightly nerdy question about from radiation oncology. So I mean, I obviously looked at dose per fraction. I mean, do you think dose rate has any effect at all? Have you looked at that? Because obviously the dose rate you get out of a a lab-based irradiator is often quite different from what you get out of a megavoltage linear accelerator.
Right, and for SBRT, the dose rate, obviously FFF versus non-FFF dose rate. We haven't looked at it extensively. We are purchasing an irradiator that will allow us to tune the dose rate. So we've preferred to do validation studies on a subset of the samples. We'll probably look at that. We'll probably look at select 30 variants that confer resistance and sensitivity, and assess the impact of dose rate. I think that's a great idea. Actually not something that occurred to me. So thanks for bringing it to our attention.
And I guess obviously, the City of London Centre, we've got quite an interest in cancer evolution. I know you're very interested in that. Well, maybe have you ever sort of done any experiments where you look for selection of these alleles within a mixed population, and what sort of frequency does that population have to be to win that game of sort of evolution in the context of radiation therapy?
Yeah, unfortunately, I don't have that slide with me, but we had published work on BRAF subclonal mutations. So we had shown that BRAF kinase domain mutations, again, the importance of this of focusing on functional parts of the gene. So if you remove the outlier associations and focus on kinase domain variants and resistance, you saw BRAF come to the top of the list. And so in that setting, in lung adenocarcinoma, those BRAF alleles were largely subclonal. Okay. And in a paper that we published in 2020 in Nature Communications, we showed that if you were to mix, so we expressed, using the S2Bs, we expressed G464V allele, which is a hypermorphic allele of BRAF, and mixed it in with wild-type BRAF. And then we irradiated. And what you saw was a substantial increase in the allelic fraction post-irradiation. And when you remove the selective pressure, when you remove the irradiation or follow the tumor over time, you saw a return back to baseline. So it suggested that there was this context-specific enrichment of a subclone that is clearly only undergoes selection under therapeutic stress.
Great, that's great. And so there's a question here from Sean. If you want to unmute your microphone and ask your question.
Hi, Mohamed. Hi, Crispin. Thanks for the great talk. Just going to ask quickly about, I mean, you've spoken a lot about KEAP1, NRF2 axis mutations and their contribution to radio resistance, and also the overlap with STK11 in terms of co-occurrence. I wonder if you've done any work to drill down on the more detail on the mechanism by which that resistance emerges, specifically thinking about the work of Gina De Nicola and the contribution of the metabolic shift and the selective advantage it might offer these tumors in terms of their ability to cope with shifts in metabolism? Or do you think it's simply a case of maintaining a more reduced environment and reducing the oxidative stress that we generate with radiation?
Yeah, it's a great question. We as a lab have not fallen down the rabbit hole of any particular gene or particular pathway. And part of that has been intentional, but it's not to minimize the importance of mechanism. We certainly think it's important. We sat on the shoulders of decades of work on NRF2, from the identification of the mutation to excellent work in the antioxidant response space that showed an increase in glutathione levels. So that was, as you alluded to, that was our initial assumption, is that the induction of a reductive state is what induces resistance. That may be simplistic. In Max's paper in Cancer Discovery later last year, he suggested that a glutaminase inhibitor can certainly reverse the resistance induced by NRF2 and KEAP1 in cell line models. That suggests that the glutathione is a major determinant of resistance, but certainly, the metabolism space is something we're very interested in. Data that I haven't shown you is we've taken the vast majority of the variants that induce resistance and conducted large-scale metabolism and analyte studies on them. And there are differences. When you look at a principal component of the different variants, they're doing different things, but they're all yet causing resistance somehow. And so that was really too fresh of data to comfortably share and relay a message of coherence. And so we're certainly interested in that aspect, both on the gene expression side as well as the metabolism side. That CYP5R4 gene is involved in mitochondrial metabolism. Clearly, metabolism is going to be key in a subset of these genes. Part of me sometimes, when I wake up in a cold sweat at night and thinking about what are the mechanisms that are causing all of this, is drivers of growth induce glutathione synthesis. And so glutathione is highly correlated with resistance across most of these variants expressed in the single cellular context. So is it as simplistic as inducing glutathione and creating a reductive capacity and mitigating the effects of oxidative stress? That's certainly a possibility for some of them, but some clearly are outliers. They're not changing reductive state and still causing resistance. I hope that answers your question. I think I meandered there for a bit.
Yeah, no, thanks. I think you're on the money in the sense that it's probably a lot more complicated, but unpicking it experimentally is really the challenge, isn't it? It's not easy to do, particularly in cell lines, obviously, with the challenges with the glutathione supplementation in the media, et cetera, that we all do. Yeah.
We found it almost intractable in diverse cancer cell lines, right? Because of all of the additional associated mutations. You might argue that this immortalized cell line context is artificial in a sense. It lacks the mutational burden that cancer cell lines, but really one of the advantages of it is you can really assess the effect of each variant in this single cellular context. So you can see how metabolic flux has changed by these distinct variants. It's a very interesting topic, and Alex Bumsicki, who's an MD PhD student, is really interested in metabolism and is going to be joining the lab in August. And so I suspect we're going to hear more from him.
Great, thanks.
That's great. Thanks so much for that question, Sean. I mean, maybe I'll just have one last question, and you probably hate this question, Mohamed. I mean, I guess obviously we know that and I'm not suggesting you have an answer or certainly any data for this, but obviously we know that there's more to tumors than just single nucleotide variants. I'm thinking particularly of copy number variants. How do you think we go about sort of analysing that sort of challenge of the effect of different copy number variants on resistance to therapy?
Yeah, well, certainly our system does overexpress. We didn't talk about the nuances of the vectors that we used, and so they're driven by a PGK and an ADH promoter. PGK tends to be a lower expressing promoter, and the ADH tends to express things a little bit higher. So we are seeing some overexpression effects. If I were to go back to that, so for example, EGFR overexpression does induce a small bit of resistance. I haven't broken it down. You know, we do have another figure here where we've separated the wild types and just show you the wild type. And so clearly we are able to measure overexpression effects. ERB2, for example, this is the wild type, not necessarily the mutant.
Okay.
And so we think we're capturing that, obviously short of doing a large CRISPR deletion collection to look at the opposite of that in terms of loss, copy loss. That's been nicely done, I think, by other groups in a cell line context. So we've decided to focus on the variants, and frankly, that's what's mostly driving biomarker selection in our clinical cohorts. Or although we do collect ERB2 amplifications, and now even in the liquid biopsy space, we're seeing their ability to detect amplifications and deletions. We think variants are higher yield, but it's not to minimize the importance of amplifications or deletions.
No, that's great. Okay, I didn't realize that you had effectively some overexpression data of the wild type allele in there. So that's
it actually made it challenging when you had a wild type that induced resistance. We had to titrate back using a different promoter to ensure that we were looking at variant selective effects. So these are nuances that I left out of here for the sake of facility, but certainly will be in the manuscript.
That's fantastic. All right, everyone. Well, I hope you all enjoyed the talk as much as I did. I'd just like to thank Mohamed for taking the time out of his busy day to talk to us all. Obviously keep your eyes out for the next list of seminar speakers. I think we've got Roger Greenberg talking at some point, as well as a host of other people. So it should be a great seminar series.